Hello, everyone, and welcome to NOAA's new Omics Seminar, NOAA's Omics Seminar Series. My name is Tracy Gill, and along with Catherine Egan, we are the webinar co-hosts for this series. Before we get started, here are a few logistics, and most are listed in the chat box. For this webinar, the presenter will allow clarifying questions, but prefers to address your questions and comments at the end of the presentation. All attendees are muted, but feel free to type your questions and comments into the chat box at any time. Also, we did have some Adobe Connect conversion problems with Sara's PowerPoint, no problem of Sara's, so we had to use the PDF, which doesn't fit the screen quite as well, but you can still see the slides. If you want to go to a full screen or larger view, look to the upper right corner of the slide deck and find the button that has a box with a bracket around it. This box button toggles the full screen view on and off. If you are interested in getting a copy of the recording, we will send them out a day or two after the talk to all who are registered. And if you're not on the weekly NOAA Science Seminar email list, but you'd like to be, please contact me at tracy.gill@noaa.gov and I will add you to the list. And many thanks to our captioner today for making this webinar more accessible. And now I will pass the mic to my co-host, Catherine Egan, the Omics Coordinator in OAR's Ocean Portfolio with NOAA's Office of Ocean Exploration. Catherine is also the subcommittee co-lead for the NOAA Omics Working Group. Catherine will introduce our NOAA Omics series, our today's topics, and our presenter. Catherine?
Thanks so much, Tracy. Hi, everyone. Thank you all for joining today. As Tracy stated, I'm Catherine Egan and the co-host for the NOAA Omics Seminar Series. So like Tracy said, the Omics Seminar Series is hosted by two NOAA groups, NOAA Ocean Exploration and the National Ocean Service through the NOAA Science Seminar Series. Omics, which is a suite of tools used to analyze DNA, RNA, proteins, and metabolites, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway both within and outside NOAA. I'm also happy to announce that the seminars from this series will now be made available on YouTube and posted up on our new Omics website a few weeks after the initial presentation. I will drop a link to our website in the chat now. It's here. So feel free to peruse our past seminars, which have been going on since June. So with that, our speaker today is Sara Schaal, and her presentation is titled Evaluation of the Role of Inversion Polymorphisms in the Evolution of Sympatric Intraspecific Diversity, a Theoretical and Empirical Study. Sara's dissertation research focuses on the genomics of ecotype evolution with a focus on the economically and ecologically important species Atlantic cod. Broadly, her research interest is in applying molecular techniques and computer simulations to understand the underlying genomic architecture of within-species adaptation. We're really pleased to say that Sara will actually be joining Dr. Ingrid Spies in Seattle to start a postdoc fellowship with NOAA this upcoming spring, where she will also be co-advised by Dr. Wes Larson at NOAA in Alaska. Once there, she will be helping to create GTC panel for Pacific cod and evaluate population structure in the Gulf of Alaska and Bering Sea using both genomics and otolith microchemistry. So Sara, congratulations on coming to NOAA, and also thank you so much for presenting today. I will now pass it off to you.
Great. Thank you so much, Catherine, for that really nice introduction. So I'm currently finishing up my PhD at Northeastern University in Katie Lauderhouse's lab, and I'm really excited to join NOAA in January. So today I'm going to present to you guys two chapters of my dissertation that complement each other. So I'm looking at the theoretical implications of inversion polymorphism. So when trying to evaluate when we would expect to find inversions in the genome that underlie adaptation. And then I also then complement this with an empirical study where I've done a field study looking at Atlantic cod in the Gulf of Maine and Iceland. And so it is actually going to be in a PowerPoint presentation, but there's going to be, we switched back to doing a PowerPoint, but there's going to be some graphs where data didn't transfer over properly. So I'm just going to try to point out as best I can what you should be seeing when it's missing. So I'll let you know when that is. So just to start with, just the importance of this work and preserving interspecific diversity is a major goal of conservation biology because species are experiencing numerous stressors in their environment. And just to take the ocean environment, because that's what I study as an example, like fishery species are experiencing unprecedented commercial fishing pressures that's specific to Atlantic cod. And we've seen their populations decline across many different populations, but they're also experiencing different stressors like climate change, where we're seeing the ocean warming to a higher degree at unprecedented rates. And especially here in the Gulf of Maine, where I study Atlantic cod, we're seeing warming that is in the 99th percentile of ocean warming across the world's oceans. So they're having to deal with a lot of stressors. So by preserving interspecific diversity, we can try to buffer these populations to have some diversity that can withstand some of these changes that they're experiencing. And just to show a study that looked at some of this type of data, looking at a conifer species, in this species, they modeled the species distribution as a single species. So in conifers, there's 3 different subspecies that are all ecologically relevant. And when modeled as a single species distribution model, just based on presence-absence data, you can see that the percent of suitable habitat over time into the future is reduced greatly compared to models that actually take into consideration the subspecies, 3 different subspecies. And you can see that that actually buffers the decline in the percent of suitable habitat. So this is just a really clear example of when this is important, especially during climate change conditions. And so within my research, I am focused on ecotype evolution. So what I mean by ecotypes is they're locally adapted populations that are genetically distinct within a species. And so these populations, they typically overlap in their habitat ranges, and they're found across many different taxa. And just to give you some examples of this, there's the resident and transient killer whales, which are 2 different they're managed separately, and their genomes are analyzed separately because there is this ecologically important divergence where they have different behaviors. And then there's also the crab and wave ecotypes of the Littorinus saxatilis intertidal snails, where they overlap in a hybrid zone in the intertidal. And finally, there's also the steelhead and rainbow trout, which one undergoes long distance migrations to the ocean to mature, whereas the other one is resident that stays in the river for the duration of its life. And so understanding this diversity and when it is expected to arise and be important for future climate change is important for managing the species as a whole and seeing when there's the potential buffering capacity of a population to climate change. And so I look at genomics of these ecotypes. And so when we're trying to identify when we would expect to see ecotypes, we're looking for regions that are non-neutral. So the divergence is concentrated in non-neutral regions in the genome. And so these areas typically are regions of low recombination. And so what I mean by low recombination is that when loci are in close proximity on a chromosome, so when they're right next to each other, they're more likely to be inherited together. So we see that that region has low recombination. And sometimes we get a bunch of loci that are all very close together on the chromosome, and these sometimes are called islands of divergence. So they come up as these selective peaks when you're looking at the genome that might be a divergence between these ecotype pairs. Or we also have inversion polymorphisms. So inversions are there's a mutation that causes the orientation of a full segment of the genome to be reversed. And these regions have really, really low recombination, almost negligible recombination, because they don't line up properly with the uninverted region. And so those inversions and anything inside those inversions are typically inherited together. And we're often seeing now, move this slide, within ecotype populations, these inversions are coming up more and more often as harboring the genetic basis for their divergence. And so this isn't a really well-known example of ecotypes, which is the annual and perennial monkey flower, so a terrestrial example. And here what I'm looking at is just looking at the genome-wide differentiation, so a structure plot. When you look across the entire genome, they look panmictic, they look very similar, but when you hone in on the inverted region in the genome and just look at, compare the annual and the perennial monkey flowers, you can see that's where the divergence is, where it lies. And so knowing that these inversions are underlying ecotype differentiation is important, and we still need better understanding of when we would expect these to arise in populations. And so the goal of my research is to take both the, like I said, theoretical and empirical approaches to understanding when we expect these inversions to arise and persist in a population. And so just to outline my talk, it's going to be two main sections, where the first is going to be my theoretical study, where I outline my simulations, where I'm looking at when the genetic basis of local adaptation is concentrated in inversions, especially in those cases where we have really high gene flow between populations, like we would expect with ecotypes. And we are asking generally, when should we expect these inversions to persist in the population and harbor local adaptation? But also, what are the typical inversion characteristics that evolve when inversions facilitate adaptation? And then I'll move into the second part of my talk, which is the empirical study, where I go over the genomic and phenotypic divergence of Atlantic cod ecotypes, both in the Gulf of Maine and in Iceland. And here I'm going to look at morphological distinctions and see if they differ between ecotypes across geographic locations, and then also whether inversions putatively underlie adaptation of these ecotypes across the populations. So looking at that first part of my talk, so I'm going to just jump into the theoretical study. This is in collaboration with my advisor, Katie Lauderhouse, but also Ben Haller, who is the creator of SLIM, which is an evolutionary simulation framework. If you haven't used it and are interested in doing any sort of evolutionary simulations, it's really a great software, and he is always open for collaborations. And so just to give a little bit of background here, oops. So to set up these simulations, I'm really just trying to understand when the genetic architecture of ecotype evolution is concentrated in those inverted regions. And so just to show you again, this is a sneak preview of my empirical study. So this is my data on Icelandic cod ecotypes. And on this left side, I'm going to be looking at the principal components of all the genomic markers in my study and seeing where our four populations fall in principal component space. So I have two coastal cod populations and two frontal cod populations, and those are the two ecotypes that they have in Iceland. And so there, when we look across the entire genome, we see that they are clustering. They are separated in a little bit in principal in PC2 axis. We have an outlier loci out here that's driving PC1, but they still are fairly overlapping in principal component space. So when we look, and this is similar to that monkey flower example, when we look at chromosome 1, and I'm looking at FST here of chromosome 1, we see these really large blocks of FST outliers, which represents a double inversion. So it's a tandem inversion. There's two inversions on chromosome 1 in Icelandic cod. And if we look at the loci in principal component space just for chromosome 1, we see that that's really what's driving the divergence on that chromosome. We see for one haplotype, so these are the different haplotypes for the inverted region, where we have homozygous for inverted, homozygous for uninverted, heterozygous, and homozygous for inverted region. And so they're differentiating the coastal cod and frontal cod within this inverted region. And empirically, across many different species now that we're getting more and more evidence about the characteristics of these inversions, we're finding that they're often large in size, so they're on the order of 5 to 15 megabases on average. And this is just, again, that example on chromosome 1 of Atlantic cod. And this is a study out of the Norwegian population where they've characterized those two inversions. And so they're often large, and they often have been estimated to be really old. So they're on the orders of millions of years old, and that's across many different species spanning marine systems to the terrestrial systems. But previous theory has had a few limitations that we're trying to basically fill in the gaps with my study. When they're trying to understand when inversions can aid in adaptation, these simulations typically require a period of allopatry or no gene flow between populations. So just to visualize what I mean by that, is there's some original population that's evolving and there's gene flow occurring. Then separation occurs that causes a barrier between the metapopulation. And now we have two populations that are adapting in isolation. At some point in these models, that barrier is removed, and then there's secondary contact between those two populations. And adaptation continues with some amount of gene flow. And I just want to focus on that word some, because a lot of these models are still really looking at low levels of gene flow between subpopulations. And that's not really what we would expect, especially in ecotypes. So that's with Atlantic cod and the Littorinus saxatilis ecotypes, the crab and wave, where they are overlapping in a hybrid zone. We do still expect there to be quite a bit of gene flow between the populations. And so theory is still, that's still a gap in the theory that I'm hoping to fill in with my simulations. And here I asked for the first question, under what conditions do inversions increase the degree of local adaptation under gene flow? And I also just, to build on this, we also, with our simulations, because we know the genetic basis of the traits that we're simulating, we can also, we can also identify when local adaptation is actually concentrated in those inversions, or when it's both in inversions and outside of inversions. So we want to see under what conditions will it really just be concentrated in those inverted regions rather than outside of them. Again, in theory, in a lot of the theory studies that have been done, inversions have been simulated as having a fixed length. So if there's more than one inversion in the simulation, it has one common length. And they also have a fixed age. So again, if there's more than one inversion in the simulation, they're introduced at a single time step. So they don't have varying ages. And so we don't have any theoretical understanding for how large inversions should be and how old they should be. And so we hope to ask that with our simulations. So when we do have an adaptive inversion, what are those characteristics? How big is it? How old is it? And the other thing that we can answer, which is an interesting debate in the literature that hasn't been resolved, is the origin of an inversion. So whether an inversion mutates in a neutral region, so there's no mutations that underlie adaptation, and then over time, does it acquire new mutations that are adaptive? Or is it another situation where there's standing genetic variation already present in the genome, and then over time, an inversion captures those loci and brings them into tight linkage, and that's what causes the inversion to eventually be adaptive in a population. And empirically, we actually have conflicting evidence. There's been two really good studies, and again, it's another study in monkey flower where ecotypes are a little bit more resolved. But in the monkey flower, their evidence suggests that the inversion does follow this capture scenario where there was already present standing genetic variation, and they used this with QTL analysis of distantly related species where these traits were present in other lineages, and then an inversion captured them specifically in the monkey flower to cause stronger divergence in this system. And conversely, in the ruff, which is a prairie bird, there's 3 different male morphs, and these morphs have different behaviors. They have different mating behaviors, different plumage colors. And these in this study, they use phylogenetics to backtrack how old the inversion is and how old these different morphs are, and found that the inversion arose neutrally, so it arose before this divergence occurred, and then eventually through mutations gained adaptive mutations over time. And this is difficult to do empirically and has only been done in a couple of systems, so having some sort of theoretical predictions would be useful to contextualize what we see empirically. So we ask, what is the evolutionary history of adaptive inversions? And finally, the last thing that I'm doing with my simulations is trying to evaluate using the methods that we use empirically, so genome scan methods, to identify whether they are accurately finding these adaptive inversions. So previous work by my advisor, Katie Lauderhouse, looked at when there was a neutral inversion in the genome, did it detect it even though it was neutral? So using different genome scan methods. And she found that if you parameterize the model with, and that takes into account linkage disequilibrium, so the linkage expected within an inversion region, you can get these models or these genome scan methods to not find a neutral inversion. But then I wanted to take that a step further, and in a genome where we have adaptive inversions and non-adaptive inversions, can it differentiate between those two? So will these genome scans find an adaptive inversion and call it an outlier, and then say that a non-adaptive inversion is a non-outlier? And so to do this, as I said, I'm using SLIM, which is an evolutionary simulation framework, and I'm using a two-population quantitative genetic model where I vary migration rate, I vary QTN mutation rate, I vary the QTN effect size and the selection strength. And what I mean by QTN is it's a quantitative trait nucleotide, so it's a single point mutation in the genome that has some effect on the trait that's evolving. And Tracy, I just want you to set up the movie now. And so just to show you some of the dynamics that happen in my simulation, here we're going to be, let me go back to the beginning. Here we're going to be looking at, I have 21 different chromosomes that I'm simulating, and that's on my y-axis here. And this is just the location along that chromosome, so the chromosome, the base pair position, so they're each 100,000 base pairs long. And I have inversions that can arise randomly throughout the genome. And this movie is showing just every 200 generation a snapshot of where current inversions are. And a lot of them are just lost due to drift or lost due to swamping from migration. But eventually what we see is we get these inversions that establish and are persisting. So those are the ones, I color them by their FST values, so how diverged they are between populations. And so we can see that there's some that arise and they persist on through the simulation. So that's just like some background to know what's happening in my simulations. Okay, Tracy, I can go back to the slides. Okay, and so I'm simulating two different patches where I have 1,000 diploid individuals within each patch, and each patch is evolving to a different trait optimum. So I'm evolving a trait that has a phenotype of -1 in one patch and +1 in the other patch. And fitness of an individual is based on how far they are from that optimum. And so walking through the times or the different steps in the simulation, I first have migration between patches. So individuals are allowed to move between the patches. And then reproduction occurs where an individual is chosen to reproduce based on how far their phenotype is from that optimum. And the phenotype is a basic additive model. So I add all those QTN mutations up with their effects on the phenotype to get their overall phenotype. And their fitness is a Gaussian fitness function. Then I generate offspring, and then I add new mutations. So then I have two different mutations occurring. One is the inversion mutations, and the other one is the QTN mutations, so the ones that are actually affecting the phenotype. And so to hop into my results, looking under what conditions do inversions increase the degree of local adaptation under gene flow? So when are those inversions actually beneficial under conditions we would expect with ecotypes? And I'm just going to take my time to orient you because the rest of the figures are going to be, for this section, are going to be in this orientation. And so on all of the x-axes are going to be the migration rate. So I did a number of different parameter combinations, and I'm going to show you all of the migration rates. So from 0.001 to 0.5, so that's low migration to high migration. Within each panel, I have 3 subpanels, which are 3 different selection strengths, which are weak selection on the trait, moderate selection, and strong selection. And then on the left side, I have a polygenic architecture. So a polygenic architecture means that I have a lot of loci, so a high mutation rate with small effects on the phenotype, and an oligogenic architecture where I have fewer, lower mutation rate, but those mutations have a higher effect on the phenotype. And so on this top panel here, the 2 rows are actually going to show the same data just in 2 different ways. And I think this is actually going to be a good thing since this is where some of the data is not going to show up. So the top graph will just show you how much local adaptation occurred in my inversion simulations. And on the bottom graph, it's going to be how much local adaptation occurred in the inversion simulation, but then subtracted from a no inversion control. And what I mean by that is for every parameter combination, I have a control simulation, so that's basically our null hypothesis for that parameter combination is these are just showing the bars are inversions that evolved, and the colored points are the QTNs that underlie adaptation. And so here you can see that the QTNs are all are mainly inside of inverted regions and have a higher FST value when they're in the inverted region. And then in the complementary, sorry, I keep forgetting to move the pointer on here. For the complementary no inversion control, what we're looking for are the cases where no local adaptation occurs compared to when inversions are present. So that means that inversions aid or help facilitate adaptation where it wouldn't have otherwise if inversions weren't present. And so here, this is just going to now show you how much local adaptation is occurring in the simulation. And down here is that difference. So what we're looking for is points that deviate from 0. And here what we see is that under those higher migration rates, so that's 0.25 and 0.4 here, we see that when inversions are present in the genome, we get elevated amounts of local adaptation. And so what that means is that inversions, here we go, I'm pointing that out, inversions do help under high migration rates, but only when selection is strong or when selection is moderate. And the moderate case, we have it only evolves under a lower migration rate. So this is really where I want to focus our results, which is that under that high migration rate where we expect ecotype migration rate to be, we see that inversions aid in adaptation when selection is strong. And this is again under the polygenic architecture. When we look at the oligogenic architecture, this is the last time I'm going to show you guys the results from the oligogenic architecture because the inversions just didn't help in adaptation under this architecture. It was really under that polygenic architecture where we see inversions aiding in adaptation. Okay, and now looking at when it's concentrated within adaptive inversions. Here, this one's going to be a little bit harder because this is where the data, the null expectation ribbon didn't plot or it didn't convert on Adobe. So here, what I'm looking at, this is just for reference. This is the same plot from before, where it's the local adaptation in inverted simulations minus local adaptation in the no inversion simulation. And there is a ribbon here that shows the null expectation for the percent of additive genetic variance in inversions. So here we're looking for the null expectation is the percent of the inverted genome. And that ribbon is, at least here, it's around here, and here it's around here. And so you'll have to trust me that these points that I'm highlighting are above that null expectation. So whenever inversions aid or facilitate the final amount of adaptation, we also see that the additive genetic variance is concentrated in inversions. And here is just an example. This is the one I showed you before. That's pretty clear that here the adaptive genetic variance is present in those inverted regions, but when they're not, inversions aren't present, there's no adaptation. But there's also all of these cases where inversions had little effect on the final amount of local adaptation, but we still had a concentration of the additive genetic variance in inversions. And what was interesting here, I'm just going to point out one example, is that in the inverted simulations, we have a concentration of loci inside those inverted regions. But when inversions aren't present, we actually were evolving islands of differentiation. So that was what I was referring to earlier in my slides, where we have islands of divergence where loci are really close together, evolving to be close together on the chromosome, so they're inherited together. So there's some conditions where inversions aren't really needed to reach the same amount of local adaptation because these islands form in the genome. And so then looking at the characteristics of the inversions, first looking at inversion age. So again, this is the same orientation of the figure where I've got weak, moderate, and strong selection and migration rate. And here I'm going to be splitting up, we binned our inversions into whether they were adaptive, non-adaptive, or whether there was no selection. So that was a control situation where we had no selection evolving, and we wanted to see how old inversions were compared to the null expectation. And here what we found was when inversions were adaptive, they tended to be much older on average compared to inversions that were non-adaptive or inversions that were present in a simulation that had no selection on it. And so just to show you what is happening in the background there for the simulation, so just for an example simulation, so this is the one that we're interested in, which is high migration, strong selection. What I'm going to show you is through time what's happening. So when each line is going to represent a different inversion, and it's going to show how the sum of all of the QTNs that are present inside the inversion and how much they affect the phenotype through time. And the bubble point is going to be how diverged it is, so the FST value. And the color is going to correspond to which population it's diverging in, so either population 1 or population 2. So if you remember, population 1 is diverging to a +1 optimum, and population 2 is to a -1 optimum. And so what we see for adaptive inversions, they establish early on in the simulation, and they persist through time, and they slowly grow, have higher FST values over time. But when you compare that to inversions that are non-adaptive, they're quite young. This is just for the inversions that made it to the end of the simulation, and you can see that they're only a couple, maybe 1,000 generations old, or a couple thousand generations old, and that's it. So they establish early and persist through the simulation. And then looking at their length, so this is the same type of plot, we see that adaptive inversions on average do have a longer length compared to non-adaptive inversions and no selection simulations. So again, this is corroborating empirical evidence where inversions tend to be large when they're adaptive, when they're found empirically. And this is just to answer that other characteristic question, which is what's the origin, the evolutionary history of the inversion? So here I'm going to be looking at 3 different what we call inversion statuses. So it's either green is when they capture, so they capture loci when they mutate and they gain additional mutations over time. They capture, the blue is going to be they capture at the time they mutate, but then they don't gain any additional mutations over time. And then the final one is that neutral scenario where they evolve neutrally, that's the purple color, and then they gain additional mutations over time. And this is going to be the average count across my replicate simulations. And it's pretty striking that whenever we had adaptive inversions evolving, they tended to always be that first scenario. So that's that capture scenario where they capture loci when they mutate and they gain additional mutations over time that harbor divergence between our populations, with just a few examples of the other two conditions occurring. And then for the FST outlier tests, I'm going to walk through the different scenarios that we looked at here. So first is whether these genome scan methods were able to call an adaptive outlier as an outlier, or whether it called it as a non-outlier. So just to show you what I mean by that, here we have two different programs. I used Outflank and PCAdapt, and again, these are inversions that evolved in the genome, and we're testing whether these genome scan methods can identify them as being adaptive peaks. And so running it through Outflank, we saw that it matched well where we see these adaptive peaks. So the red means that they were called as outliers, and the black means they weren't called as outliers. But then we did see a couple of cases where PCAdapt would call an adaptive inversion non-adaptive. So this was false negative in this case. And the next we looked at the non-adaptive inversions. So those are the inversions that are evolving in the genome that don't harbor loci underlying the trait under selection, so they're non-adaptive. And we wanted to assess how often they were called as potential outliers, or if they were called correctly as non-outliers. And so here in this case, we have one example where we had some concentration of loci that made it it was called as an outlier, even though it was a non-adaptive inversion in Outflank. And so just summarizing that across our parameter space, we have Outflank on the right, PCAdapt on the left, and the colors that you want to pay attention to. So the green means they did it correctly, so adaptive outlier and a non-adaptive outlier. And the darker colors, the blue and the purple, are when these programs called a non-adaptive inversion as an outlier or an adaptive inversion as a non-outlier. And really, I'll just say overall, these did really, really well, better than we thought they were going to do, except there was just a few conditions, especially in Outflank, where we had more non-adaptive outliers when migration rate was really low under strong selection and moderate selection. Finally, when we looked at a no selection simulation, so there's no selection occurring, we wanted just to make sure that these weren't called as outliers. And we did actually see a few instances in PCAdapt where even though none of these loci were under selection, we did see some inversions being called as outliers. So these would be non-adaptive called outliers. And we can see that across our parameter space. It's really only Outflank that has this happen. I'm sorry, PCAdapt that has this happen. But it's really, again, only a few instances of this. So we're pretty confident that overall these methods work for finding differentiating between adaptive inversions and non-adaptive inversions. So in summary for this chapter, or this section of my talk, chapter of my dissertation, we show that inversions do aid in adaptation under high gene flow, and this occurs when the trait is polygenic. So in our case, there were thousands of SNPs that underlie the trait that was diverging. And when selection was strong for that trait. We also identified the conditions when sometimes, even though inversions did aid in adaptation when they were present, if they weren't present, we still had, the genome sometimes did evolve these islands of divergence that would bring the population to a similar amount of local adaptation when they weren't present in the genome. And so we've identified with our simulations these transition periods when the islands of divergence can evolve, but then there are the higher gene flow scenarios that I showed previously, where only when inversions are present could they aid in, or could the population evolve any amount of local adaptation? And compared to empirical studies, we have corroborated the studies that show that inversions are old and large. So all of the adaptive inversions in my simulation tended to be very old, and they typically were larger in size compared to our null expectations. And interestingly, they almost always capture standing genetic variation at the time of mutation. So that's that capture origin of the inversion. And like I said previously, overwhelmingly, these outliers methods seem to work at differentiating between inversions that are adaptive and inversions that are non-adaptive. We occasionally get a few false positives under a couple of scenarios, but we think that those are fairly minor. Okay, now I'm going to move on to the second part of my talk, which is the characterization of the genomic and phenotypic divergence of Atlantic cod in the Gulf of Maine and Iceland. And this is a collaboration between John Grabowski in my department, Graham Sherwood at the Gulf of Maine Research Institute, John Puritz, who was a postdoc in our lab, and Shobanas Secker, who is works at research computing at Northeastern. And so looking at my time, I'm going to go through this introductory stuff a little bit quick. But here I just wanted to give you some background on the population. So in Iceland, as I alluded to earlier, we have a frontal ecotype and a coastal ecotype. So frontal are offshore cod that undergo long distance migrations, sometimes all the way to Greenland. And coastal cod that live near shore and live in coastal regions year round. And they tend to be larger when they're coastal cod and prefer structured habitat, whereas the offshore cod tend to be a little bit smaller and have no habitat preference. And some genomics have been done on these populations to show that there's the 4 inversions that are present that differentiate the ecotypes, and this has been done in Iceland and Norway, where we have linkage group 1, linkage group 2, 7, and 12 showing inversions that are present in the genome. And when we look in principal component space, they overlap quite a bit. We have some divergence of the coastal cod in Norway from the rest of the group. But we can see these large outliers when we bin them by their migratory behavior. And this was from Berg et al. But no study so far has linked their traits to ecotype divergence. So looking at the morphometric differences between the 2 ecotypes and linking it to the genomics. And most of the work has focused on the southwest population of Iceland, with the ecotypes in Iceland, where we add in a second population where we're looking at also the northeast corner of Iceland. We want to know whether inversion haplotypes are present in both populations. And in the Gulf of Maine, it's a little bit more complex, where we have coastal cod that are red in their coloration, and we also have an offshore cod that is olive. So there's been a lot of work looking at these 2 color morphs, where their body shape tends to be less streamlined when they're coastal cod and live in coastal structured habitats. And the olive cod are more streamlined and have no habitat preference. So they have a stouter, deeper body with in the coastal regions, and then more slender tails as well. And that's been work done by Graham Sherwood and John Grabowski. And from one of John's students, Chris Conroy, he showed that also these cod have shown preference to different thermal habitats. So red cod can withstand temperatures, or they stay present in temperatures throughout the year. So this is throughout an entire year, and these are the summer months when the temperature is starting to warm. So this is temperature down here. And we see the presence of the 2 color morphs, where red cod tend to persist longer in the year compared to olive cod. So there might be some temperature adaptation between these color morphs. But we also have a spring and winter spawning group in the Gulf of Maine, where we have where recent work has shown that there is the presence of these potential inversions on linkage group 7 and 12, but also a few outliers on linkage group 18 and linkage group, oh, sorry, that was linkage group 11. And so we wanted to see where red cod fit in this picture with the spring and winter spawning groups. So we wanted to look, is there a genetic basis driving the color morphs, and can we link this to the traits of interest? So they're morphological traits. And so I already went through this, so I'm just going to speed up ahead a little bit in the interest of time. And so this was our sampling regime. We collected cod from Iceland on trawl surveys from the coastal region and the offshore region in 2 locations, the southwest and the northeast. And in Massachusetts Bay, I collected red and olive cod from Cash's Ledge, which is an offshore seamount where we see these red and olive cod living together in sympatry, and also from Massachusetts Bay. And we tried to bin these into the 2 different spawning groups post hoc, and we only could confidently bin 16 individuals as winter spawners. And we had 40 that were unknown, so they were kind of between spring and winter spawners. So for the genomics analysis, and all further analyses, I'm really comparing this group of 16 to the group of 33 red cod. And these, we collected fish via hook and line or trawl. We imaged, weighed different aspects of the fish. We removed an otolith and stored a fin clip for genetics. And for the morphometrics, we used box truss measurements. So we used 11 landmarks. We removed the landmark, which was landmark 11, which has been biased due to gut fullness. So any measurements coming from that point were dropped. Oh, and we did a linear discriminant analysis for understanding the different morphometrics. For sequencing, we did whole genome sequencing on a NovaSeq paired end lane. We did 15x coverage, and we sequenced 296 samples. We looked at principal components to look at structure. I've been exploring some of the haplotype structure to look at interesting regions in the genome and comparing them across populations. And then we did FST outliers using outflank. So first, morphometrically, I'm just putting the different morphometrics that loaded on our discriminant functions. This first one, I'm not going to go through the morphometrics that differentiate these. I'll talk through the ones on the next slide, but this was comparing all populations together. We see that it's kind of a mess. They're all very similar, but we do clearly see that the red triangles here, which represent coastal Icelandic cod, and the blue triangles here, which represent offshore Icelandic cod, are differentiating from each other. And so that becomes quite clear when we split it up into the 2 different locations. So this is just Icelandic cod. We see that they differentiate from each other in discriminant space, and the loadings represent the offshore cod having a longer tail and a stouter face. So their tails are more streamlined, but also the coastal cod have deeper bodies, so they have those stouter bodies that we would expect of a non-migratory fish. Whereas the Gulf of Maine cod, we're really seeing the separation occur. So again, the colors represent the 2 different color morphs. The olive cod, we're on that first discriminant axis, we're seeing it split by population. So this is Cash's Ledge and Massachusetts Bay. And we're seeing that a lot of the measurements are located on the face. And so they have stouter faces at Cash's Ledge, which probably will have to do with how they feed on the seamount. And on the second axis, we do see that there is some evidence for the red cod having stouter bodies and shorter tails, but it's not strong. And so when looking at the genomics, I'm just going to quickly go through this. We see them, when I look at all populations, we see them split by region, geographic region, where we have Iceland here and the Gulf of Maine here. When we hone in on each of those regions individually, here's the Gulf of Maine. When I look across the whole genome, they look fairly panmictic. There's no clear groupings there. However, in Iceland, we do see some separation between our two different ecotypes, so the offshore cod and the nearshore cod. And is also clear when we do the genome scan method, the genome outlier detection methods. We see these 4 major inversions that are present both in the southwest Icelandic cod population and the northeast Icelandic cod population. So we show that those haplotypes are present in both populations. They are diverging between the 2 ecotypes in the way that we would expect. But when we compare this to, sorry, that's what I just said. When we compare this to the Gulf of Maine, we see very few outliers. So I kept these bars in just to show you where the inversions were present in the Icelandic cod, but we see there's no outliers that are present in these regions. We have just a couple of outliers, sparse outliers, throughout the genome in Massachusetts Bay, but no outliers present in Cash's Ledge. And what's interesting here is, I'm just going to skip past this, I'll go to this one here. On chromosome 2, if we look at the principal components of the genotypes on chromosome 2, we see the inversion is actually present in both the Gulf of Maine and in Iceland. Iceland is where we expect it, and it's diverging between the ecotypes. But it's not diverging between our color morphs. So the points are just a jumble of all the different populations. So the inversions are present, they're just not diverging with color morph. And that's the same for chromosome 7 and 12. And so just in summary, no variation in body morphology is corresponding to color morphs in any meaningful way. It's very minor, which suggests that we may not have a migratory ecotype in the Gulf of Maine. And we also have inversions present, but they're not segregating with those color morphs. And then in Iceland, we have body morphology indicative of a migratory cod, which is what we would expect because they do undergo long distance migrations, and the 4 large inversions present. And with that, I would like to thank a ton of people for all of their help in the field, for computational assistance, and for all my collaborators and my advisory committee, and my funding. And we already talked about this, but I'm going to be working on Pacific cod at NOAA in Seattle and Alaska. And I'm happy to take questions.
Thank you so much, Sara. Incredibly interesting presentation. Like I said, we're really happy to have you joining NOAA. Looks like we have a question coming in from Krista. Color morphs, are they associated with diet in this species? Perhaps you mentioned it?
Yeah, so actually diet is interesting because we've had some conflicting results. 2 different studies now have come out. One suggested that the red cod was eating more lobster and crabs, so getting a diet that's that's different from the offshore cod, which is feeding mainly on pelagic fish. But then we had another study, another student worked up some data from the same populations, but didn't find a difference between their diets. So this is something that's still a little bit contentious, and we're not sure if there is some association with diet. So we still need some more research on that aspect to know for sure.
Thanks, Sara.
Thanks for the question.
Okay, we have another question in. We have another question coming in from Diana. Great talk, Sara. Is it correct that the most important life history characteristic that influences whether adaptation is found in inversions or islands of divergence is whether the population has high migration rates or gene flow? Are there others?
So that's one aspect, and so the thing, so gene flow had to do with it, but also the strength of selection. So we show that when migration rate is high, so that's in my simulations that was a 0.25 or 0.4 migration, so that's a lot of mixing between populations. The only way that there could be adaptation in those populations is when there was inversions and when selection was strong. So there is this interesting aspect of selection playing a role in whether inversions can aid in adaptation. And that's kind of what we would expect, especially with ecotypes, is that selection is probably very strong for those traits to diverge when they're living in sympatry. So there's some niche that's in that sympatric distribution that they're evolving. So selection is probably very strong on the trait.
All right, and now we have a question coming in from Tracy. Could climate change be affecting migration and thus inversions?
So that's another really interesting question. That's why we were thinking that it would be interesting to see inversions in the Gulf of Maine, because it is warming so quickly, like they're experiencing warming conditions currently. And so finding an inversion that might underlie some sort of thermal adaptation could help us delineate different populations that may more or less be susceptible to climate change. But we didn't see that in the color morphs. So we did see it in Iceland, but Iceland is actually going to experience a period of warming that's actually going to be beneficial to cod in that region. So we're going to see populations increase. And so this is actually something I'm working on. It's not going to be a chapter of my dissertation, it's going to come afterwards. But I'm doing otolith microchemistry to associate the temperature that these cod live in Iceland to their genome to see if these inversions are related to any thermal adaptation that might be expected in these populations. And that'll be a direct link that we can talk about what might happen with climate change.
All right, questions are coming in. So we have a question from Mo. Great talk, Sarah. Maybe I missed it, but in the Gulf of Maine, are the red and olive cod entirely sympatric, or do they sort into spawning groups separated by timing, winter versus spring, or spawning location?
Yeah, I'm so sorry I had to go a little fast over that part. I wish I would have had more time. I did that a little slow. My first part a little slow. So there's spring and winter spawners that have been identified in the olive cod, but we don't really understand where red cod fits in that picture. And so that's kind of what I was trying to maybe get a handle on with the genomics, is whether they would if they would group more with a spring spawning cod or a winter spawning cod. But because we didn't find many spring spawners in our sampling, we couldn't associate whether red cod were a part of one of the spawning groups. And just from my collaborator Graham Sherwood, he's told me when he's been out at these spawning aggregates, he doesn't see red cod. So we're not really sure what when they are spawning, and there hasn't been a lot of work to know whether red cod are spawning with winter or spring. And so we were hoping with my study we'd find that out, but unfortunately we didn't have spring spawners in my sampling to know for sure.
Great, thank you, Sarah. Because we only have a couple minutes left, we'll just do one more question, but I encourage you all to please reach out to Sarah with your additional questions here. So we'll take this last one from Richard. Will your findings affect Atlantic cod assessment or management in the Gulf of?
That's a good question. My guess is probably not, because these aren't managed. So currently, color morph isn't considered a separate management group at all. And because I'm not really I haven't showed any any striking difference genomically in these two groups to think that there's some diversity that's harbored within the red morph versus the olive morph that's worth preserving. We're still going to look at the thermal tolerance, so I'm still going to look at what temperature water they live in across their life, because we do think that's probably different in the coastal cod versus an offshore cod. So the coastal cod will experience a wider range in temperatures throughout the year compared to offshore cod. But currently with my data, I don't think that this gives any evidence to assist in changing any management strategies in the Gulf of Maine right now. And it's been notoriously difficult to change the stock boundaries in the Gulf of Maine, and there's a bunch of groups working on that right now with a lot of other data. And the spring and winter spawning groups are more of where they're going to try to tackle the management aspect.
All right, thank you so much, Sarah. We are at time, everybody, so I'm going to just quickly wrap up. Want to thank you again, Sarah, for coming to speak at the Omics Seminar Series. Very fascinating presentation, and thanks to everybody for joining us today to listen in. Just a reminder that the NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time. Oh yes, and thanks to our capture as well. Thank you, Tracy. Our next seminar will take place on November 17th, 2021, and is titled Connecting Microbes, Phytoplankton, and Oceanography in the California Current with eDNA and Metagenomes. And this will be Dr. Nastasia Patton with NOAA AOML. Registration will be up soon on the NOAA Omics website. And with that, thank you all again, and have a wonderful rest of your day.
Thank you.